Diseases that run in families have been known for many years. These can be non-life threatening, like albinism, or result in premature death, like Huntington's disease. We've already used each of these as examples of recessive and dominant traits in humans. In the remainder of this chapter, we'll be discussing some of the genetic disorders that have been found in the human population. First of all, sickle cell anemia. We've referred to it a few times already, but it's the first human disease that was shown as a result of a mutation in a protein. It's caused by a defect in the oxygen carrier molecule hemoglobin. Hemoglobin is classically made up of 4 domains, or 4 different subunits, 2 alpha subunits and 2 beta subunits. One of the beta subunits has a mutation so that it forms an improper hemoglobin molecule. Forms sort of a rod-like structure instead of a 4-dimensional structure, and it causes them to stack up inside the red blood cells and causes them to sickle like so, instead of maintaining their regular shape like these puffy ones over here. Of course, if there's not enough space on the hemoglobin, it's going to lead to impaired oxygen delivery to the tissues. Not only that, but these sickled cells have a multitude of effects, including getting stuck in the smallest of blood vessels, causing impaired circulation, and many other features. Any individual that's a homozygote for sickle cell allele, that is, they carry this recessive allele on both chromosomes, will have intermittent illness throughout their life, as well as a reduced lifespan, simply because of these circulatory problems and oxygen carrying capacity. Heterozygotes, though, appear normal at normal altitudes and oxygen levels. They do have hemoglobin, but only half as much as necessary, and so they have reduced ability to carry oxygen only when demands are maximal. So when an individual who's heterozygous for sickle cell moves up to, say, our altitude, a lot of the effects of sickle cell anemia will appear for them just because of the lower perfusion of oxygen. This same individual, if they were to be a marathon runner, would also probably have some pretty profound effects from the sickle cell allele. The sickle cell allele is particularly prevalent in people of African descent, which is an interesting situation. In fact, up to 45% of the population could be heterozygote, which is much higher than would be expected if this was random distribution. It turns out that the heterozygosity actually confers greater resistance to the blood-borne parasite that causes malaria. And so it's actually an advantage for people in areas where malaria is prevalent to have the sickle cell allele, because one stage of the malaria parasite requires human red blood cells for its development. Without those human red blood cells, the malaria can no longer wreak havoc on the system. That would explain why there's a much higher frequency of that allele in certain populations. Some other conditions that we might consider are a result of nondisjunction. Nondisjunction is the failure of homologs or sister chromatids to separate properly, either during meiosis I or during meiosis II. What this results in is having a gamete that has 2 copies or no copies of a particular gene. When that gamete comes together with the other gamete, it'll result in either having 3 copies of a chromosome, like you see in this karyotype here, or only 1 copy. These are called aneuploidies, the gain or loss of a whole chromosome. Monosomy is when we just have one of those chromosomes, so we lost one to nondisjunction. And trisomy is when we gain one. In most cases of nondisjunction, the zygote produced does not survive. However, some of the smallest autosomes that you can see here can be present in 3 copies and allow the individual to survive. Like if we look at chromosome 13, 15, 18, 21, and 22, they can survive. Although 13, 15, and 18 cause some severe defects and the babies will die within just a few months. 21 and 22, however, can survive into adulthood. Trisomy 21 is Down syndrome. The majority of trisomies result from a full third chromosome, so nondisjunction usually occurring in the egg in this case. It could also happen, though, from just a part of chromosome 21. That is, if there's a translocation of one piece of chromosome 21, like maybe the chiasmata don't separate completely properly, and the little piece of chromosome 21 ends up in the baby, then this can also produce the effects of Down syndrome. Now, naturally, the mother's age is going to influence the risk in nondisjunction disorders. This is because a woman has all the eggs that she's ever going to have at the time of birth, and they're all stalled in meiosis 1. They don't actually complete meiosis 2 until the event of fertilization. So her eggs are as old as she is. And the mechanisms, like the spindle apparatus and so forth, are old also. And so they're much less effective at carrying out the final stages of meiosis. Here's a few statistics to consider. In mothers younger than 20 years of age, the risk of having a Down syndrome baby is about 1 in 1,700. In 20 to 30-year-old mothers, the risk is still only about 1 in 1,400. In 30 to 35-year-old mothers, the risk rises to 1 in 750, and by the age of 45, the risk is higher than 1 in 16. This is why it's so important to have screenings done as an older mother to make sure that many of these nondisjunctions aren't there. You can also note that the majority of miscarriages have to do with nondisjunction for any of these chromosomes shown here. The body can simply detect that something's not quite right. Sometimes the mechanisms of detection are better than others. Individuals who gain or lose sex chromosomes do not generally experience any severe developmental abnormalities. They have somewhat abnormal features, but often reach maturity and in some cases may even be fertile. For example, if an XX gamete combines with an X gamete, the result is 3X zygote. This develops into a female with one functional X chromosome and 2 bar bodies. She may be a little bit taller in stature, but otherwise appears relatively normal. And if an XX gamete combines with a Y gamete, then the effects are a little more serious. The resulting XXY zygote develops into a male who has many female body characteristics, including breasts, and in some cases, but not all, a diminished mental capacity. This condition is called Klinefelter syndrome. If an O gamete, that is one with no X or Y, fuses with a Y gamete, the resulting 0Y zygote is non-viable and fails to develop any further. It will be miscarried. But if that O gamete fuses with an X, we have an XO zygote and it develops into a sterile female. It's just missing bar bodies. These females will be of short stature and have a webbed neck, and the sex organs never fully mature during puberty. Their mental abilities are in the low to normal range, and the condition is called Turner syndrome. We could also see Y chromosome nondisjunction. In this case, you have the formation of a YY gamete. When these gametes combine with an X gamete, we have an XYY zygote, which develops into a fertile male of normal appearance. And there's a number of studies that were performed on males that were in prison, and they found a lot of XYY individuals. This was back in the 70s. They thought these men to be super males, and they were particularly aggressive and angry and so on and so forth. But when you look back to their teenage lives, you recognize they had more acne than the average male of their age, and thus they were probably outcasts. And this can certainly result in some anger and delinquent behaviors. So this just goes to show that correlation does not mean causation. In the late 20th century, geneticists really thought that they understood the basic mechanisms that govern inheritance. And it became quite a surprise when some geneticists studying mice found that there were some important exceptions to classical Mendelian genetics, and it appears to be unique to mammals. This is genomic imprinting. where a phenotype that's exhibited by a particular allele depends on which parent contributed the allele to the offspring. So if the father contributes the allele, it may be expressed differently than if the mother contributes the allele. Some genes are inactivated in the paternal germline, and others are inactivated in the maternal germline, and therefore they're not expressed in the zygote. So it essentially makes the zygote haploid for the imprinted gene. An example of genomic imprinting in humans involves two diseases. There's Prader-Willi syndrome and Engelmann syndrome. The effects of PWS include respiratory distress, obesity, short stature, and mild mental retardation, and some obsessive compulsive behavior. In AS, we see developmental delay, severe mental retardation, hyperactivity, and aggressive behavior, as well as inappropriate laughter. The most common cause of both of these syndromes is a deletion of the material on chromosome 15. In fact, it's the same deletion causing either syndrome. It just determines whether it's of paternal origin or maternal origin, whether the child gets Prader-Willi or Engelmann's. Prader-Willi syndrome arises if the chromosome deletion is paternally inherited, that is, from the father, and it's Engelmann syndrome if it comes from the mother. Genomic imprinting's not that well understood. At least one aspect seems clear, though. The basis for inactivating genes appears to be linked to modification of the DNA itself. DNA can be modified by adding methyl groups, or it can become methylated. This modification is correlated with inactivity of the genes, so highly methylated areas of the DNA are inactivated. Bar bodies are a great example of highly methylated X chromosomes, and thus, these methylated regions affect gene expression of that region. Although most genetic disorders cannot be cured. We are learning a great deal about them, and great therapies are being developed in many cases. Without having any cures, the only resource is to try and avoid production of children with these conditions. The process of identifying a parent's risk for having children with these genetic disorders and assessing the genetic state early in embryos is called genetic counseling. This can consist of pedigree analysis, where we use a family's history to determine the probability of genetic disorders in the offspring. We can also use amniocentesis, which we can use to collect fetal cells from the amniotic fluid. It takes a number of weeks for examination because you have to culture the cells and wait for them to grow, and there are some implicit dangers in doing this. It could cause rupture of the uterus and thus miscarriage of the fetus. There's a newer technique that's been developed called chorionic villi sampling. It's much less invasive, and we insert a needle to take some of the chorion, which is the fetal side of the placenta. We examine these cells. We can tell a lot about the fetus. The more that we know about the Human Genome Project and possible abnormalities in genetics, the more we can screen using this chorionic villi testing. So as time goes on, the stronger our capacity is to identify abnormalities early in pregnancy. The chorionic villi testing can be done much earlier than the amniocentesis, so it's also a preferable technique for that reason. We can detect these disorders early in the first trimester. Let's take a quick card quiz. Bar bodies are all genes, nuclei, mitochondrial, and chloroplast genes are inherited from both parents. Trisomy 21 is known as... What chance will a daughter have of being colorblind if she has a normal mother and a colorblind father? This one may take a moment. A woman who is colorblind reproduces with a man who has normal color vision. Their sons will... Here are your answers.